Well, hello, everyone. For this week's Life Science Lab, we've got a lot of microscopy. Because last week we talked about general trends in plant diversity, and we spent a lot of time in the greenhouse looking at different types of plants' morphology, how they look different, how they grow differently. This week we're going to take a lot of those different plants and look at them under the microscope. So, to properly conduct this lab, you should be looking at these images and then pausing the video regularly as different samples are going to be needed to be drawn in pretty good detail. So the lab video will be relatively short because I want you to spend some time drawing what you see and paying attention to the different tissues. So let's go ahead and get started. Based on the lab instructions, which you certainly should have read prior to watching this video, the first leaf that we're going to be looking at is the lilac plant, or syringa. Now, lilac has some features that we're going to want to identify. The epidermis, or the skin being on the top and the bottom of the plant, leaf. The mesophyll, of which there are two different layers, the palisade and the spongy. Conductive tissue. We'll talk more about that later in this lab, but generally the veins of the plant. The cuticle, the waxy layer on top of the conductive tissue. And then the guard cells, or stomata, which is the opening on the bottom side of the leaf, and the airspace. So let's take a look at what this lilac leaf looks like underneath the microscope. To recreate what you should be doing in lab whenever you're focusing the microscope, you always should start at 40x, the farthest away magnification. Here you see the lilac leaf furthest away. You see that there are different colors visible, some blues, some purples, and some pinks. Those different colors are going to correspond to different cell wall biochemistries, meaning that they're made of different things because those are different types of cells. So we're going to be looking at cell coloring to help identify where different internal tissues are. Next up, we would have focused on the leaf at 100x. We start seeing more definition between the different cell layers. And then finally, we get to our 400x, zooming in on one of the closer vascular bundles. Now, looking at the different structures we're supposed to label, we of course have our epidermis, the skin cells on top. On top of the epidermis itself, you will occasionally see some waxy layer, a waxy surface or waxy shading. That is the cuticle, so of course we label that. Going down into the tissue, you notice that there is some cells that are very tall. These are part of the mesophyll or middle leaf, and the ones that are tall kind of look like a fence, being our palisade mesophyll. Beneath the palisade mesophyll, we have our spongy mesophyll. And you notice how there are much bigger air gaps in this tissue. Now those gaps are on purpose and present pretty much everywhere, those air spaces on the underside of the leaf, because we want those undersides of the leaf connected to the mouths of the leaf. That was the epidermis, but rather the mouths are the stomata, and you can see a stomata here, the opening being present right there, and then two guard cells on either side. You also have two guard cells here with the stomata here. So you see that there are air spaces and places for air to penetrate because the leaf is going to want to breathe in that CO2. You guys remember that from photosynthesis. The last section, last unlabeled feature, is our conductive tissue, which is kind of a mess here, but we'll take a look at it in greater detail next. Now, of course, you should pause the video here and draw what you see, labeling all of these different tissues. Next up, we're taking a look at two different types of plant stems. the simple xylotum and the more complex sunflower. Now, you could potentially label everything you need to see at 40x here, but I think it's a little bit more appropriate to zoom a little bit further down so that way we can see everything. Now, the reason why xylotum is more simple is because it has a singular vascular bundle, singular conductive tissue in the middle. There are no multiple strands. The multiple strands of conductive tissue would suggest that this plant makes leaves, and xylotum does not. Hopefully you do remember it from last week. It was our plasticky looking plant. And kind of one of my favorites because it looks so archaic, so old. But under our microscope, we can see the different tissues that comprise this. Now, of course, we have our epidermis, the outer layer. You do, in fact, see some of the stomata present on that epidermis, even though this is a stem. You have the ground tissue, or the different tissues that make up the storage or photosynthetic organs. And then you have the different parts of the conductive tissue. The singular vascular bundle in the middle, with a light green layer called the phloem that transports sugar, and the pink stuff, xylem, that transports water. So please take a moment to go ahead and draw this simple structure. Next up, we'll take a look at our sunflower stem. From 40x, we again see that it is much different, much more complex than our xylotum stem. And most notably, we see lots of vascular bundles and even some weird pink tissues up top, some supportive tissues. And we still have a huge amount of ground tissue here. One major notable difference is that in xylotum, the xylem makes up the very middle of the stem. In plants that are more complex, like angiosperms, we oftentimes encounter a pith region, something in the center that differentiates our simple plants from our more complex ones. Multiple vascular bundles, as well as presence of a pith. Looking at the sunflower stem in higher resolution, we see again that there's an epidermis, we see again that there is ground tissue, and we see again that there is pink xylem, greenish phloem, and then unlabeled here we have this pink strand of supportive tissue, all comprising our conductive tissue. So go ahead and draw both the xylotum and the sunflower at 100x. For our next drawings, we need to take a look at our monocot and dicot flowers. But let's go ahead and take a moment to take a look at the dicot flower model. Hey everyone, so here we are with our flower model. Ready to learn some things about a flower model? So, flowers are very unique. They're part of the reproductive structure for the angiosperm lineage, which from our lab last week, hopefully you remember that is one of the most derived lineages of plants, and flowers are really their defining feature. So what is a flower? What are the components of a flower? Well, drawing back to one of our early plant biologists and philosophers, Goethe, in German, he very eloquently said that everything ends up being a leaf. And he was right. All of these components of a flower are actually derived from a leaf. Leaves in a flower grow in something called whorls, or rings. And so if you're looking at a flower facing down, you can see that there is a whorl in the middle, a whorl just outside of it, a petal whorl, and then underneath the petal whorl, a sepal whorl. So you have those four layers. The infertile sepals, which are the green leaves underneath the flower, right? The next whorl, get this back in there, are oftentimes colored petals, and just like a leaf, they have veins, but
They are colored this time instead of green and photosynthetic. So the petals are our second whorl. After those two whorls, which are both considered the infertile whorls, which means that they don't actually go on to make babies. Petals are just showy. Sepals are just energy producing. We have our two reproductive whorls. Males, then females in the very middle. So it's always the female whorl that's at the very center, the very heart of the flower. The male whorl is defined by two features. The filaments that are essentially long stalks with the spore or pollen producing thing at the tip. This is called the anther. You're always going to have pollen made in these structures. The number of pollen sacs that you get in the anther is actually derived by whether that plant is a monocot or dicot. So think back to our floral buds and think back to how many pollen sacs those different flowers had. Our last central whorl is the female house, the female circle. And for the purpose of this class, y'all need to remember two components to the female circle. The ovary itself, the heart of it, which has the eggs inside, and then the extended part called the style with the stigma or landing pad for pollen on the very top. And the way reproduction happens in a flower is that pollen will be spread, oftentimes by some sort of pollinator like a bee, onto the stigma. It'll land on the stigma, and then it begins to grow its way through, and literally grow and eat its way through the female house. It's kind of weird. But it will find its way to the egg where fertilization will happen. And so hopefully that gives you an idea of the complexity of a flower. Yes, everything derives from a leaf, and this is a 4-layered leaf where you've got sepals, petals, male, female. But all of this works in unison to help attract pollinators, spread genetic diversity, and create fruits.
All right, so now that you know the parts of a basic flower, the different whorls, let's go ahead and see if we can find differences in the patterns behind how these whorls are constructed between monocots and dicots. Now from lecture, you'll hopefully remember that monocots are our organisms with parallel veins, flowers in parts of 3, and are oftentimes thought of as our grasses. Our dicots are instead looking for flower parts in series of 4s or 5s. So here we'll start with a monocot, and you'll notice that the very middle here, that female ovary is building ovaries in parts of 3. This triplicate pattern is also observed in this secondary monocot flower bud, where again there are 3 chambers growing here. Again, ovary in the middle, female home in the middle, male home, all of our different anthers on the outside, and then behind that we see the beginning development of petals and sepals behind that. Can't really differentiate sepals and petals up until they've started to make flower colors, so under the microscope they look fairly similar. So these are our monocots in parts of 3. Looking at our dicots, we see that they grow things in parts of 4s and 5s. 1, 2, 3, 4, 5. 5 sets of anthers that are each going to be grown on their own filaments. Looking from a greater distance, you can again potentially see the sets of 3s or 6s, 1, 2, 3, 4, 5, 6 on that monocot. A little bit harder to see from this distance on the dicot flower bud. But if you zoom in, you can see again that the dicots on the left, monocot on the right for this comparison of our two different tissues. Zooming in, beautiful parts of 3s and 6, and zooming in, beautiful sets of 4s and 1, 2, 3, 4, 5 for our dicots.